Welcome to the deep dive. Today, we're tackling a subject that might seem a little in the weeds at first glance: embryology.
Yeah, they can sound pretty dense.
Right. Specifically, we're going to explore the incredibly detailed process of how your head, neck, and the inside of your mouth actually took shape.
Hmm.
Now, even if you think this sounds like dusty textbooks, our aim is to pull out the really crucial and surprisingly fascinating bits. The kind of knowledge that gives you a real understanding of how things work.
Like a backstage pass.
Exactly. Think of this as your personal backstage pass to the development of a pretty important part of you.
That's right. And we've got some rich material to work with. We've been looking at a video lecture titled One Embryology AMP4 and also a corresponding PDF document, oneembryology.pdf.
Both packed with info.
Absolutely, both packed with information. Our goal in this deep dive is really to synthesize the key insights from both of these sources, giving you a clear and connected understanding of this complex process.
Exactly. Our mission today is to truly grasp the fundamental steps involved in building the structures of your head, your neck, and, well, everything within your oral cavity.
And teeth.
And teeth. We're particularly excited to unravel the amazing story of how your teeth develop, and importantly, to connect any hiccups in that early development to the dental conditions we actually see in the real world.
Makes sense.
Okay, let's get into it, starting right at the very beginning.
So if we look at those very early days, around the third and fourth weeks of embryonic development, this is when the absolute foundations are being laid.
The groundwork.
Precisely.
Mhm.
During the third week, a pivotal event occurs called differentiation. A single layer of cells, known as the epiblast, it sort of reorganizes itself into three distinct layers: the ectoderm, the mesoderm, and the endoderm.
Okay.
For you, think of this as the very first blueprint of your body. Each of these layers is, destined to give rise to all your different tissues and organs.
So getting that right is crucial.
Absolutely.
Yeah.
If something were to disrupt this fundamental organization, it could unfortunately have pretty widespread consequences.
Okay, so week 3 is this critical sorting into 3 foundational teams, let's say. What are the major events then in week 4 that start to build on this initial organization?
Week 4, it's an incredibly dynamic period. Gastrulation, the process of forming those germ layers, that actually continues.
Right.
Then we see neurulation, which is the formation of the neural tube, the very early precursor to your brain and spinal cord.
The beginning of the nervous system.
Exactly. Alongside this, the embryo begins to segment into repeating units, and crucially, the whole structure starts to fold in on itself.
Folding, like origami.
Huh. Sort of, yeah. These might sound like abstract concepts, but these early processes are absolutely essential for establishing the basic layout of the entire body.
So from these initial layers and this flurry of activity, we start to see more specific structures emerging, right? That leads us to the branchial arches. I think our sources also called them pharyngeal arches.
Yes, both terms are used. Pharyngeal is maybe a bit more precise for humans.
Okay, pharyngeal arches. This sounds like a key step in actually constructing the head and neck.
It is indeed. The pharyngeal arches are essentially embryonic structures that appear and will eventually develop into a significant portion of your adult head, neck, and even the upper part of your chest.
Wow.
The term bronchial actually has roots in the Latin word for gills, which, hints at our evolutionary history.
Right, like fish.
Exactly. But, as I said, in human anatomy, pharyngeal arches is generally the preferred term. These arches, along with another structure called the frontonasal prominence.
That forms the forehead and nose, you said.
That's the one. Together, they're really responsible for sculpting your lower face and neck. So think of these arches as the early construction crew for your face and neck region.
Okay, so these arches are the master builders here, but what are they actually made of? It sounds like those early germ layers we talked about, ectoderm, mesoderm, endoderm, they each contribute.
That's a great question. Yes, each pharyngeal arch is a result of contributions from all 3 primary germ layers we just discussed: the endoderm, the mesoderm, and the ectoderm.
And something else, too.
And, importantly, derivatives of neural crest cells.
Ah, neural crest cells. They come up a lot in development, don't they?
They really do.
Yeah.
They're a fascinating population of cells, unique to vertebrates, actually. They migrate extensively during development and contribute to a really diverse array of structures.
Like what in this case?
Well, parts of the skeletal system in your head and neck, like bones in the skull and jaw, parts of the ear, cartilage, plus components of your peripheral nervous system. Their ability to move around and differentiate into so many different cell types makes their proper development particularly critical.
Wow, so it's a real team effort at the cellular level. It makes sense then that if these pharyngeal arches don't develop correctly, the sources mention potential defects.
Unfortunately, yes. If the pharyngeal arches don't form as they should, it can lead to a variety of anatomical and developmental defects. These can manifest in many different ways, depending on which arch is affected and the specifics of the developmental error.
It really highlights just how crucial these early, precisely coordinated events are.
Absolutely. Timing is everything.
And it seems like timing is also really critical here. The sources specifically mentioned pharyngeal pouch development happening around the 4th week, too.
Yes, the formation of the pharyngeal pouches, which are essentially inward bulges or pockets of the endoderm lining the early pharynx, occurs around the same time as the arches are developing, in that crucial 4th week.
Pouches inside, arches outside-ish.
Kind of. The pouches are internal invaginations, and they're incredibly important because they give rise to a number of vital structures in your head and neck region.
So we have these arches forming externally and then these pouches appearing internally between them. What exactly is the structure of one of these pharyngeal arches themselves? Like, what's inside?
Okay, so each pharyngeal arch has a central core of mesoderm.
The middle layer.
Right. This mesoderm will eventually differentiate into the muscles, nerves, arteries, and skeletal elements, like cartilage or bone, that are associated with that particular arch.
Okay.
This mesodermal core is then covered on the outside by ectoderm, the outer layer, and on the inside by endoderm, the inner layer.
Got it. Layered.
Exactly. This layered organization allows each arch to contribute specific components to the developing head and neck in a very organized way.
Okay, a layered structure with a mesodermal heart. Now, the sources also talked about pharyngeal clefts and pharyngeal pouches, one coming from the outside, one from the inside. What's the key difference and what do they eventually become in us?
Good distinction. The pharyngeal clefts are essentially inward folds or grooves of the ectoderm on the outer surface of the embryo, located between the arches.
Outside grooves.
Right. The pharyngeal pouches, as we mentioned, are similar inward folds, but of the endoderm on the inner pharyngeal side.
Inside pockets.
Exactly. As for what they develop into,
Yeah.
the first pharyngeal cleft is quite significant as it forms your external auditory meatus, your ear canal.
Ah.
Interestingly, the second, third, and fourth pharyngeal clefts don't really persist in their original form. They typically come together to form a temporary structure called the cervical sinus,
Cervical sinus.
which normally just disappears during development. It regresses.
So those outer grooves mostly vanish, except for the one that becomes our ear canal. What about those inner pouches then? What do they turn into?
The pharyngeal pouches have some really important fates. The first pharyngeal pouch develops into the primitive tympanic cavity, which is part of your middle ear, and the auditory tube, you might know it as the eustachian tube.
Connects the ear to the throat, right?
Exactly, connects your middle ear to your nasopharynx. The second pharyngeal pouch gives rise to your palatine tonsil, a key part of your immune system in your throat. The third pharyngeal pouch has a bit of a journey, eventually forming your inferior parathyroid glands and your thymus gland, both crucial for hormone regulation and immune function.
Thymus and parathyroids from the third pouch.
Inferior parathyroid specifically. And finally, the fourth pharyngeal pouch develops into your superior parathyroid glands.
Superior parathyroids from the fourth. Wow.
It's quite remarkable how these seemingly simple early structures give rise to such diverse and vital adult components.
It truly is amazing. Now, the sources also broke down the fate of each of those primary germ layers, endoderm, mesoderm, ectoderm, specifically within the branchial pharyngeal arches. What's the general contribution of each in this head and neck region?
Okay, generally speaking, the endoderm lining the pharyngeal arches primarily develops into the endocrine glands of the neck.
Like thyroid.
Exactly. This includes vital glands like your thymus, thyroid, and parathyroid glands, all playing critical roles in producing and regulating hormones.
Got it. Endoderm makes glands. What about mesoderm?
The mesoderm within the arches gives rise to the vasculature, so the blood vessels and the muscles of the head, neck, and upper thorax.
Muscles for chewing, smiling.
All of those, yes. The muscles that allow you to make facial expressions, the muscles you use for chewing, for example.
And the ectoderm.
And finally, the ectoderm has a couple of important roles here. One part, the neural ectoderm, contributes to various components of the nervous system in this region. The other part, the surface ectoderm, forms the skin.
Skin and nerves.
And importantly, as we touched on earlier, the neural ectoderm also gives rise to those crucial neural crest cells,
Ah, them again.
which then contribute to the bones of your skull, your jaw, your ear, the cartilage in your head and neck, and parts of your peripheral nervous system. So each of those initial layers has its own specialized role in building this complex area.
Okay, so each layer has its job. Now let's get into the specifics of each of the first four and maybe those rudimentary fifth and sixth pharyngeal arches. This is where we can really see exactly what each one contributes. Let's start with the first arch, the mandibular arch.
Right. The first pharyngeal arch is a real workhorse, especially for your lower face. Its pharyngeal cleft, as we discussed, forms your external auditory canal.
Ear canal, check.
The arch itself contributes to your mandible, your lower jawbone, as well as two of the tiny but crucial bones in your middle ear that help you hear, the malleus and the incus.
Hammer and anvil bones.
That's them. It's also responsible for the muscles of mastication, the chewing muscles, and the maxillary V2 and mandibular V3 branches of the trigeminal nerve.
Big nerve for the face.
Huge. It provides sensory information from much of your face and oral cavity, plus motor control to those chewing muscles. The first pharyngeal pouch, remember, develops into your eustachian tube and the mastoid air cells in your temporal bone.
Right.
And the nerve supply directly associated with this first arch is those maxillary and mandibular branches of the trigeminal nerve, CNV.
That's a significant list from just the first arch. Okay, moving on to the second arch, the hyoid arch. What does it build?
The second pharyngeal arch, or hyoid arch, contributes to the stapes, the third small bone in your middle ear, the stirrup.
Malleus, inasus, stapes. Got it.
It also forms the body and the lesser horn of your hyoid bone.
That floating bone in the neck.
That U-shaped bone, yeah, that supports your tongue. This arch is also responsible for all the muscles of facial expression, the ones that let you smile, frown, wink.
Ah, facial nerve territory.
Exactly. It's innervated by the facial nerve, cranial nerve 7th, and the second pharyngeal pouch, as we discussed, develops into your palatine tonsil.
So, second arch. Facial expressions, stapes, part of the hyoid, tonsil pouch, facial nerve. Okay, now for the third arch, what's its role?
The third pharyngeal arch contributes to the rest of the hyoid bone, the body, and the greater horn, as well as the superior constrictor muscle, which is involved in swallowing.
Swallowing muscle.
Yep. It's also associated with the development of the internal carotid artery. The third pharyngeal pouch is quite busy, forming your thymus, the piriform recess in your laryngopharynx, and your inferior parathyroid glands.
Thymus and inferior parathyroids again, right?
And the nerve of the third arch is the glossopharyngeal nerve, cranial nerve 9X. It has roles in taste, swallowing, salivation.
CNIXS. Okay, we're building up a really detailed picture. Let's talk about the fourth arch now.
Okay, the fourth pharyngeal arch. This one is responsible for the main cartilages of your larynx, including the thyroid cartilage, the Adam's apple part of your voice box.
Voice box structures.
Right. It's innervated by the vagus nerve, cranial nerve X, specifically the superior laryngeal branch. This arch also contributes to the aortic arch and the right subclavian artery, important vessels, as well as various muscles of the larynx.
Wow, arteries too.
And in the fourth pharyngeal pouch gives rise to your superior parathyroid glands.
Superior parathyroids. Fourth pouch. Got it. And finally, we have the fifth and sixth arches, which the sources mention are a bit different, maybe rudimentary.
That's right. The fifth pharyngeal arch is often considered rudimentary, maybe merges with the sixth, doesn't contribute significantly on its own to adult structures that we clearly define. The sixth arch is closely associated with the fourth.
So what do the fourth and sixth together do?
Together, these arches contribute to the inferior pharyngeal constrictors, more swallowing muscles, and other intrinsic muscles of the larynx. They are innervated by the vagus nerve, CNX, specifically the recurrent laryngeal nerve branch, and maybe some input from the accessory nerve, CNXA.
Okay.
The pharyngeal pouch associated with these later arches, sometimes called the ultimobranchial body, gives rise to the parafollicular C cells of your thyroid gland.
Pure C cells, they make calcitonin.
Exactly, the hormone involved in calcium regulation.
That's an incredible amount of detail. It really highlights the complexity. Now let's shift gears a bit and dive into another fascinating story. The life cycle of a tooth. This seems like a whole other intricate process unfolding within the structures we've just discussed.
It absolutely is. Tooth development, or odontogenesis, is a long and precisely orchestrated sequence of events. It's typically divided into 6 main stages: initiation, proliferation, histodifferentiation, and morphodifferentiation, apposition, and finally calcification or maturation.
6 stages. Sounds complex.
Each stage is critical. and relies on really complex interactions between different cell types.
Okay, let's start at the very beginning then, with initiation, also known as the bud stage. When does this kick off, and what's happening at the cellular level to start forming teeth?
The very first signs of tooth development can be seen quite early, actually, around the 6th week of embryonic life.
Week 6. Wow.
At this stage, cells in the basal layer of the oral epithelium, that's the lining of the developing mouth, they start to multiply more rapidly than the surrounding cells.
Okay, rapid division.
This increased cell division leads to a thickening of the epithelium in the region where the future dental arches will be, sort of extending along the future jawline. This thickening is called the dental lamina.
Dental lamina, the start line.
Pretty much. It's considered the foundation for the ectodermal part of the teeth, specifically the enamel. At the same time, in each developing jaw, about 10 small rounded swellings or buds appear along this lamina.
10 per jaw, baby teeth.
Exactly. These correspond to the future primary teeth, the baby teeth. Interestingly, the permanent molars, your adult back teeth, will also arise directly from this dental lamina a bit later on. But the permanent incisors, canines, and premolars actually develop as buds sprouting off from their primary tooth predecessor's buds.
So it's like a first wave for the baby teeth, and then the adult teeth, except for the molars, kind of branch off from that initial structure.
Fascinating. What kind of problems can arise during this very early initiation phase? Well, errors during initiation can lead to some relatively common dental issues. If the initiation process doesn't happen correctly, or if there's an early halt in cell multiplication, proliferation.
Things stop too soon.
Right. It can result in the congenital absence of one or more teeth, meaning you're born without them, anodontia or hypodontia.
Missing teeth.
On the other hand, if there's continued or excessive budding from the dental lamina, it can lead to the development of supernumerary teeth, extra teeth beyond the normal number.
Too many teeth. Okay, so initiation is all about laying down the initial groundwork. What happens in the next stage, proliferation, also known as the cap stage?
Okay, the proliferation stage, which occurs around the 9th week of development, is characterized by continued and often uneven growth of that initial tooth bud.
Uneven growth.
Yeah, this unequal growth leads to the formation of a cap-like structure, which is why it's called the cap stage. A shallow indentation appears on the deep surface of this cap. The cells on the outer and inner surfaces of the cap will eventually form important layers called the outer and inner enamel epithelium.
So it's growing and changing shape.
Exactly. And just like with initiation, problems with proliferation can lead to issues with the number of teeth. Too little proliferation can result in a tooth germ failing to develop altogether, leading to a missing tooth.
Again, missing teeth.
Or, if there's too much proliferation, it can lead to the formation of epithelial rests, small groups of cells that kind of get left behind. Sometimes they can become active later, potentially forming cysts.
Cysts from leftover cells?
Potentially, yes. Furthermore, if some of these proliferating cells become partially specialized, maybe detached from the main structure, they might start producing enamel and dentin in a disorganized way.
Leading to
the formation of odontomas, which are a type of benign dental tumor or even more supernumerary teeth.
So the cap stage is this period of rapid growth and shaping, and errors here can have diverse consequences. Okay, now we move into the Bell stage, which you said is divided into histodifferentiation and morphodifferentiation. That sounds like a critical phase for defining the tooth's actual identity and shape.
It absolutely is. The Bell stage, starting around the 11th week, is when the enamel organ continues to invaginate, taking on a distinct bell-like shape, hence the name.
Makes sense.
This stage is really crucial because it involves both histodifferentiation, that's the process where cells become specialized for their specific jobs.
Like becoming enamel makers or dent makers?
Precisely. And morphodifferentiation, which is the process where the basic form and size of the future tooth are established. Morphodifferentiation essentially lays out the blueprint for the future junction between the enamel and the dentin, the DEJ, and it relies on continued cell multiplication.
Okay, so shape and cell specialization happening together.
Right. In the early part of the Bell stage, histodifferentiation really kicks into gear. The cells of the dental papilla, that's the mesenchymal tissue, nestled inside the bell of the enamel organ, start to differentiate into odontoblasts.
Odontoblasts make dentin.
Correct. These are the cells that will form dentin, the bulk of the tooth. At the same time, the cells of the inner enamel epithelium differentiate into ameloblasts.
And ameloblasts make enamel.
The enamel formers, yes, the hard outer layer of the crown. Interestingly, this histodifferentiation marks the end of the major proliferative phase for these specific cells. They largely lose their ability to multiply significantly after this point. They're committed.
Okay.
And this cellular specialization is driven by a fascinating back-and-forth signaling process called reciprocal induction.
Reciprocal induction, that sounds really important. Can you break that down for us? How does that work?
Certainly. Reciprocal induction is this crucial communication system between the developing enamel-forming cells and the dentin-forming cells.
Hmm.
It works something like this. The preamyloblasts, which are the inner enamel epithelium cells before they fully become enamel producers,
Okay, preamyloblast.
they send a signal to the adjacent cells of the dental papilla, instructing them to differentiate into odontoblasts, the dentin-forming cells.
Signal 1 to become odontoblasts.
Right. These newly formed odontoblasts then begin to secrete the dentin matrix, the organic framework of dentin. Once this initial layer of dent matrix is laid down, it then sends a signal back to the preamyloblasts.
Signal 2. Okay, I'm making dentin. Your turn.
Exactly, causing them to fully mature into a ameloblast and start producing the enamel matrix. So it's a continuous loop of signaling that ensures the coordinated and sequential formation of these two hard tissues. It's really elegant.
Wow, that is elegant, like a little cellular dance.
It is. And alongside the inner enamel epithelium, we also have other important layers of cells within the enamel organ at this Bell stage. The stratum intermedium and the stellate reticulum.
Supportive layers.
Essentially, yes. They play a supportive role in nourishing the ameloblasts and facilitating enamel formation. Now, notably, the outer enamel epithelium, the outermost layer of the enamel organ, has a different primary role later on.
Not enamel formation.
No. It's involved in the later stages of tooth eruption and the formation of the junctional epithelium that's the soft tissue seal between the gum and the tooth.
Okay, different job for the outer layer. What about the bottom of the bell?
Ah, good point. The cervical loop, that's the area where the inner and outer enamel epithelium meet at the rim of the bell. This gives rise to a structure called Hertwig's epithelial root sheath, or HERS.
Hers sounds important.
Absolutely crucial. HERS determines the shape of the tooth root, and significantly, it lacks that stellate reticulum layer we just mentioned.
Which
Which means no enamel forms on the root. That's why roots are covered in cementum, not enamel. Remnants of HERS can sometimes persist as small clusters of cells near the root, known as the epithelial rests of molasses,
leftover root-shaping cells.
basically. Meanwhile, cells of the dental sac, or dental follicle, which is the connective tissue surrounding the developing tooth germ, come into contact with the newly formed predentin on the root surface.
And they become?
They differentiate into cementoblasts, which will form cementum, the outer layer of the tooth root. This cementum can be cellular, meaning it contains trapped cementoblasts, or acellular, without trapped cells.
Okay, that's a really detailed picture of the Bell stage. And that reciprocal induction mechanism is fascinating. It sounds like if that communication breaks down, it could lead to some specific problems. What kind of defects might we see arising from issues during histodifferentiation or morphodifferentiation?
Exactly, because histodifferentiation involves the specialization of cells to produce either enamel or dentin. Errors at this stage can lead to defects in the structure of these tissues.
Like faulty materials.
Precisely. A classic example of ameloblast failure is amelogenesis imperfecta, which results in abnormally formed enamel. It can be thin, soft, pitted, easily worn, or discolored.
Affecting the enamel quality.
Right. Similarly, if the odontoblasts don't differentiate or function properly, it can lead to dentogenesis imperfecta, characterized by abnormal dentin structure that's often weaker, more translucent, and prone to wear.
Affecting the dentin quality.
Correct. Now, in terms of morphodifferentiation, which establishes the tooth's shape and size, disturbances during this phase can result in variations in tooth morphology.
Like weird shapes.
Exactly. Things like peg teeth, small, cone-shaped teeth, often laterals, or microdontia, where teeth are significantly smaller than normal, or macrodontia, where they're larger than normal.
Size and shape problems. Okay, so the Bell stage defines the cells and the shape. What happens next in the apposition stage?
The apposition stage, kicking off around the 14th week, is all about the layer-by-layer deposition of the extracellular matrices, the scaffolding of enamel and dentin.
So the actual building starts.
Yes. The ameloblasts and odontoblasts, now fully differentiated and specialized, line up along that future dentino-enamel junction, DEJ, and on the root side, the dentinocemental junction, DCJ. They begin secreting their respective organic matrices, enamel matrix by the ameloblasts and dentin matrix, predentin, by the odontoblasts.
Laying down the framework.
Exactly. This deposition follows a very precise pattern and occurs at specific rates, originating from distinct growth centers, like cusp tips or incisal edges, and progressing outwards and downwards.
And if this gets interrupted?
Good question. If anything disrupts the function of the ameloblasts during enamel matrix formation at this stage, Maybe a systemic illness in the developing baby, high fever, nutritional deficiency, or even local trauma.
Like an infection.
Yes, like an infection affecting a developing permanent tooth bud from an overlying infected primary tooth that can lead to enamel hypoplasia.
Hypoplasia. Less enamel.
Exactly. It's a defect in the amount or thickness of enamel formed. It might look like pits, grooves, or areas of missing enamel. That example of local trauma causing it is often called a Turner's tooth.
Turner's tooth. Okay. What about dentin?
Dentin hypoplasia, a defect in the amount of dentin formed, is less common. It usually requires a more significant systemic disturbance during development to affect dentin apposition.
So apposition is the actual building of the tooth structure framework, layer by layer. Finally, we reach the calcification maturation stage. What's happening here to give the tooth its final hardness?
Right. Calcification, also known as mineralization, is the crucial process that follows the deposition of the enamel and dentin matrices. It involves the precipitation of inorganic calcium salts, primarily in the form of hydroxyapatite crystals, within those deposited organic frameworks.
Soaking the framework in minerals.
Essentially, yes, which hardens the tissues dramatically. This mineralization process typically begins at the tips of the cusps or the incisal edges of the teeth and progresses cervically towards the root. It involves the formation and eventual fusion of small, spherical, mineralized areas called calcospheroids.
Little balls of mineral fusing together.
Kind of like that, yeah. The time frame for the completion of calcification varies. It takes about 2 years for the crowns of primary teeth to fully mineralize after they start, and around 4 to 5 years for the crowns of permanent teeth.
Takes a while.
It does. And if this calcification or maturation process is disturbed, for example, if the calcospheroids in dentin don't fully fuse together, it can result in a microscopic defect within the dentin called interglobular dentin.
Gaps between the mineral balls.
You could think of it that way. Other defects that can occur during maturation include enamel hypomineralization or hypomaturation. Here, the enamel matrix is laid down okay, but it doesn't fully mineralize to its proper density.
So the right amount, but not hard enough.
Exactly. It results in softer, more porous enamel that can appear chalky, opaque, or discolored, and it may wear down faster. Conditions like dental fluorosis, caused by excessive fluoride intake during development,
Fluorosis. The white spots.
can be white spots, or brown staining in more severe cases, and also tetracycline staining that happens if a mother takes tetracycline antibiotics during pregnancy, or if a young child takes them while teeth are developing. These also fall into the category of maturation defects affecting calcification.
Staining built right into the tooth.
Unfortunately, yes.
Okay, that's a really comprehensive overview of the tooth life cycle, from that initial thickening of the oral lining all the way to the hardened, mature tooth. It's incredible to think about all the coordinated cellular activities involved. The sources also briefly mention the different embryonic origins of the dental tissues. Can you give us a quick rundown of where does enamel come from versus dentin, pulp, etc.?
Absolutely. It connects back to our earlier discussion. Enamel has a unique origin. It's derived from the enamel organ, which itself originates from the ectoderm, one of those primary germ layers.
Ectoderm makes enamel.
Correct. Dentin and the dental pulp, the soft tissue inside the tooth containing nerves and blood vessels, both originate from the dental papilla. This is a type of embryonic connective tissue called ectomycin.
Ectomesenchyme. Neural crest cells involved.
Yes. Importantly, this tissue has significant contributions from those neural crest cells we talked about earlier.
Ah, okay. Neural crest cells are key for dentin and pulp. What about the root structures?
Cementum, the hard outer layer of the root, the alveolar bone, which is the bone socket that surrounds and supports the teeth, and the periodontal ligament, or PDL, the fibrous connective tissue that attaches the tooth root to the bone. All three originate from the dental follicle, also called the dental sac.
And that's also
Derived from ectomesenchyme, with neural crest cell input. So it's a mix of the initial ectoderm germ layer and those migrating neural crest cells that gives rise to all the tissues of the tooth and its supporting structures.
fascinating. Neural crest cells really are multitaskers. Now, the sources also highlighted some key morphological differences between primary baby teeth and permanent teeth. What are some of the main distinctions someone might notice?
Yeah, there are several notable differences in shape and structure. For example, the crowns of primary teeth tend to be wider from side to side mesiodistally relative to their height, their crown length, compared to permanent teeth. They look a bit more squat sometimes.
Wider, shorter-looking crowns.
Generally, yes. The roots of primary anterior teeth, like the incisors and canines, are typically narrower and longer, in proportion to the crown.
Skinny, long roots on the front ones.
hmm The roots of primary molars are also generally longer and more slender than those of permanent molars, but they tend to flare out more towards their ends, mesiodistally,
Flared roots on the back baby teeth. Why?
to make space. They need to accommodate the developing permanent premolars underneath them before they fall out.
Ah, makes sense.
You might also notice a more pronounced cervical ridge, that bulge of enamel near the gum line on the lip and tongue sides of primary anterior teeth.
A bigger bulge at the neck of the tooth?
Yes. And the crowns and roots of primary molars are often more constricted, or slender, at the cervical third, near the gum line. The buccal cervical ridge, the bulge on the cheek side, is particularly defined, especially on the upper and lower first primary molars. It's quite distinct. Also, the chewing surfaces, the buccal and lingual surfaces of primary molars above that cervical bulge are often flatter, which results in narrower occlusal surfaces, narrower tabletops, compared to permanent molars,
Flatter sides, narrower chewing top.
generally. And finally, a very obvious difference is color. Primary teeth are typically lighter and whiter in color than permanent teeth, which often have a slightly more yellowish or grayish tinge, sometimes called milk teeth for that reason.
Lighter color, that's easy to spot. Were there any specific primary teeth mentioned as being particularly unique?
Yes, a few stand out. The upper central primary incisor is unique in that it's usually wider mesiodistally than it is long incisor-cervically, the only incisor like that. The upper primary canine has a very sharp cusp, and its root often leans towards the back of the mouth, distally. The lower central primary incisor has a pretty straight cutting edge, and its root is usually about twice as long as the crown. The upper first primary molar, looks a bit like a premolar, has a very prominent cusp on the tongue side towards the front, mesiolingual cusp. The lower first primary molar is quite unique, often has two distinct cusps on the cheek side, buccal cusps, and the chewing surface narrows a lot towards the tongue side. And the primary second molars, both upper and lower, tend to resemble the permanent first molars in their overall shape, just smaller.
Those are some really helpful and specific points to keep in mind. Okay, finally, the sources briefly mentioned a tooth eruption table. Is there anything key we should know about that in the context of development, besides just the dates?
Yes, absolutely. There is a typical and fairly predictable sequence and time frame in which both the primary and permanent teeth erupt, or emerge, into the oral cavity. While memorizing specific dates isn't the main point here, it's important to understand that this eruption sequence is another critical aspect of dental development.
The order matters too.
Exactly. Deviations from this normal pattern of eruption, for example, teeth erupting much earlier or later than expected, or in an unusual order, or if symmetry is way off between left and right sides, can sometimes be an indicator of underlying developmental issues, nutritional problems, endocrine disorders, or other medical conditions. So while we focused on the formation of the teeth themselves, their emergence into the mouth is also a key part of the overall developmental picture and can give clues about health.
Right. Another piece of the puzzle. This has been an incredibly deep dive, really, into the world of embryology, specifically zeroing in on the development of the head, neck, oral cavity, and our teeth. It's truly amazing to see just how precisely orchestrated and interconnected all of these processes are.
It really is, starting from those very early embryonic stages.
Just a few cells, essentially.
Absolutely. It's remarkable how these intricate steps, occurring so early in life, lay the fundamental groundwork for the complex structures we have as adults. And understanding these developmental pathways isn't just, an abstract scientific pursuit.
No, it connects to real life.
It provides crucial insights into the origins of various congenital anomalies, birth defects, and dental conditions that individuals may encounter later in life.
Yeah.
By understanding what normally happens, we gain a better understanding of what can, on occasion, go wrong and why.
Definitely. For you, the listener, what were some of the aha moments or surprising facts that really stood out during this deep dive? We've covered a huge amount of ground.
From those three initial germ layers,
To the pharyngeal arches building the face,
the intricate signaling that builds our teeth, that reciprocal induction.
even the differences between baby and adult teeth.
And when you really stop and think about the sheer complexity and coordination required for all of this, it raises some fascinating questions, doesn't it?
Like what?
Like what other seemingly subtle variations during embryogenesis, maybe tiny timing shifts or slight molecular differences, might have subtle, long-term effects on our health or anatomy that we don't yet fully understand? It's a field where there's still so much to explore and discover.
That's a really thought-provoking point to consider, the long-term echoes of these very early events.
Hmm.
So if anything we've discussed today has sparked your curiosity, we strongly encourage you to delve deeper into specific aspects. There are a wealth of resources available on developmental biology, the genetics of dental anomalies, the clinical side of embryological defects.
Absolutely. Ultimately, it's truly awe-inspiring to reflect on the inherent ability of the human body to orchestrate such an incredibly intricate and precise developmental program, all starting from just a few initial cells.
It really is a
Yeah.
powerful testament to the elegance and complexity of biology.
Well,
Well said. Thanks for joining us on this deep dive. Until next time, keep exploring the amazing world around and within you.